Hi, my name is Alexander Sousa, and I am a graduate student researcher in the lab of David Liu at the Broad Institute of MIT and Harvard. Two research areas in the Liu laboratory are the development and application of biological tools that can make precise changes or edits in DNA. We call these tools precision gene editors, and we can use them to rewrite DNA like a text file in a word processor. Using precision gene editors, we can correct errors in DNA that cause disease by rewriting the individual letters that make up DNA. My research in the Liu laboratory is focused on developing a precision gene editing strategy to correct the DNA mistakes that cause AHC, and also to understand how fixing these mistakes at the DNA level can reduce or eliminate symptoms associated with AHC. The goal of a future gene editing therapy for AHC would be the correction of AHC mutations in patients to prevent AHC disease. To achieve this goal, I am developing precision gene editing strategies that correct the exact AHC-causing mutations that are found in patients. In collaboration with the Swedner lab at Mass General Brigham and the George lab at Northwestern, I'm evaluating the potency of these correction strategies in cells from AHC patients. By developing potent editing strategies in patient cells, I am moving towards the first step of developing a future AHC gene editing therapy. In collaboration with the Lutz lab at Jackson Laboratories and the Swedner lab at Mass General Brigham, I am also using mouse models of AHC to evaluate how gene editing can correct the symptoms of AHC. Mouse models allow us to test drive gene editing treatments that inform future AHC therapies. With these AHC mouse models, we can see how using gene editing to correct the AHC DNA errors can relate to reduction or reversal of AHC-related symptoms in a living organism. These insights will inform the design of future AHC therapies. Without a doubt, the enthusiasm of the AHC patient community for science and their existing network of clinicians and scientists that could support our own research inspired us to take on an AHC gene editing project. It's been a delight to work with the AHC patient community. Their broad knowledge base and the connections that they have made it really easy to make the choice to work on AHC for our own gene editing projects. I was very lucky to present at an AHC conference recently, where I met with researchers, clinicians, and families in the AHC community. While there, I had a chance to meet with many AHC parents and hear about the challenges that they and their children face every day dealing with AHC. I found the resilience and positivity in the face of this devastating disease as an inspiration to do the best research that I can to uncover insights that might lead to a future therapy for AHC. If I could share a message with AHC families, I would say that we are working as hard as we can every day to uncover scientific insights needed to move closer to a gene editing therapy for AHC, and that we're thinking of them every step of the way.